Hello friends, in this video today we will be discussing about recombinant DNA technology. As the name suggests, it is based on the concept of the gene recombination. That is, we are combining a gene from one organism to some vector and then we are incorporating it into some another organism. So the definition encompasses a number of experimental protocols leading to the transfer of genetic information, which is a DNA, from one organism to another. It involves the manipulation of genetic material to achieve the desired goals in a predetermined way. The present day recombinant DNA technology has its roots to experiments performed by Boyer and Cohen. Indeed, genetic engineering, which could be considered as recombinant DNA technology, is a modern technology which was which is recently a very important boom for the biomedical sciences, research sciences, agriculture, and has a very large impact on modern medicine right now. Since it basically involves the manipulations at the genetic level, it has a crucial role in human life. The basic principles of recombinant DNA technology, that is rDNA technology, involves the generation of DNA fragments and selection of the desired piece of DNA, then insertion of the selected DNA into cloning vector to create a recombinant DNA, which is also known as chimeric DNA. It involves the introduction of recombinant vectors into host cells, multiplication and selection of cloning containing the recombinant molecules, expression of the genes to be produced the desired product. So we know production of recombinant DNA involves the incorporation of a specific DNA or a gene of one segment, which is cleaved and just transferred to a bacterial plasmid, which is rather transferred to a particular host. For example, like a briefing in this figure, a DNA segment is used, it is then ligased into a vector, the vector is then implanted in a specific transformed cells, then by cell division, mitosis, meiosis, the cell further multiplies and the same DNA gene is expressed into a huge number of cells. Restriction endonucleases play a crucial role for the manipulation of DNA. Talking about them, they are the bacterial enzymes that plays an important role in cutting or splitting the DNA to specific sites. Initially, they were discovered in E. coli, restricting the application of bacteriophages by cutting the viral DNA. The host E. coli DNA is protected from cleavage by addition of methyl group. Generally, initially E. coli was, as postulated says, the bacteriophages, the viruses that kills bacteria. So the bacteria have developed a tendency to break down the viral DNA with the help of restriction endonucleases. Next come the recognition sequence, as it is not possible to it is not worthy to clean out or cut the DNA at any sites. So recognition sequences are a crucial important role. Inside, they are the sites where the DNA is cut by restriction endonucleases. Restriction endonucleases can specifically recognize DNA with a particular sequence of 4 to 8 nucleotides and cleave. Cleavage pattern, the cut DNA fragments by restriction endonucleases may have a very most sticky ends or blunt ends. The DNA fragments with sticky ends are particularly useful for rDNA experiments, since single-stranded sticky DNA ends can easily pair with any other DNA fragments having complementary nature. Talking about restriction enzymes, they are type 1, type 2 and type 3 restriction enzymes, depending where they are doing the cuts, whether it's near to the desired gene or far away from the desired gene. You can get through this different bacteria through which restriction enzymes can be isolated, like bacterial amylophysiums, Brevibacterium albidum. So there are numerous number of bacteria for the same. A general phenomenon of restriction enzyme, how it cuts and cleaves the DNA and the formation of sticky ends. The sticky ends then leads as you can see, the sticky ends formation, sticky ends leads to the site where a complementary other strand could be attached to it. While a restriction endonuclease also very essential, since it helps a restriction endonuclease can also produce blunt ends. See, there are no sticky ends, so we can say these are the blunt ends. DNA ligase, on the other hand, has a very important role in attaching the desired DNA segment. So, if we are having cleaving of the DNA and we are not able to attach it to a specific site, then it is not at all of any use. So, DNA ligase are the enzymes that are very important and bind the desired cut DNA to a specific DNA, the site of interest. After the attachment, now it becomes very essential to which organisms can be used. Since the desired DNA has been manipulated, it is not having the DNA of just one organism or one species. It is very essential to transfer the desired or the specific recombinant DNA into a specific desired host. The host must be competent enough. It is very essential. If the host is not competent and it is not able to sustain the recombinant DNA, that whole of the process, the whole of the experiment becomes useless, since the recombinant DNA will not be able to replicate itself into the host, and no further multiplication, and whole things goes in vain. So host cell could either be a prokaryotic or a eukaryotic cell. Microorganisms are preferred as host cells since they multiply faster as compared to cells of higher organisms. So indeed, microorganisms, that is bacteria or fungus, have a very important role, and they are preferably used with comparison to others. E. coli was the first organism used in the DNA technology experiments. The major drawback is that it cannot perform post-translational modifications. So as you can see throughout the figure, as previously said, a specific foreign DNA of interest is having cutting site. The same sticky ends for that desired gene is present in a vector which we are using, and if it is matched, the sticky ends matches, it is attached, then it is being transferred to a host which could be a bacterial chromosome, and as the bacteria divides, the same will happen with the recombinant DNA which you are having, that is the recombinants. Eukaryotic hosts, these are the preferred to since when we talk about medical application of recombinant DNA, so it becomes very essential to create the recombinant proteins or the development of the recombinants which are eukaryotic cells. So these are preferred to produce human proteins since they have complex structure. Mammalian cells produce the machinery to modify the proteins to active form, leading to post-translational modifications, for example tissue plasminogen activator. Talking about vectors, they are the DNA molecules which carry a foreign DNA fragment to be cloned. Most of the vectors are plasmids, bacteriophages, cosmids, and artificial chromosomes. Like talking about plasmids, they are the extrachromosomal double-stranded circular self-replicating DNA molecule. Usually plasmids contribute to about 0.5% to 5% of the DNA of bacteria. A few bacteria contains linear plasmids. The plasmids carry genes resistant. That is a very important thing. That if it provides a help, it provides a boom for the selection process. That if a gene which is transformed, which is added to a desired vector, and the vector is having the selective markers which could be ampicillin or tetracycline, it provides easiness in the selection processes. When we talk about bacteriophages, they are the viruses that can also be used vectors since they have a high replication state and they can easily sustain in bacteria since having a good affinity and infection causing property, for example phase lambda phase and M13 phase. Cosmids are the vector possessing the characteristics of both plasmids and bacteriophage lambda. These carry fragments for foreign DNA compared to plasmids. Artificial chromosome vectors, they are the recently developed chromosomal vectors that, like example human artificial chromosome, YAC, yeast artificial chromosome, bacteria, they can accept large fragments of foreign DNA. Methods of gene transfer. The uptake of plasmid DNA by E. coli is carried out by ice-cold CaCl2. Transformation occurs in the presence of CaCl2, in which the pores are made and the transformed can be that is the recombinant DNA can incorporate within the cell. Conjugation is a natural microbial recombination process in which the desired recombinant plasmid having the DNA is transferred to the attached cells. Electroporation is based on the principle that high voltage current, when a high voltage current is provided to a specific cellular system, it creates some pores, and the pores enable the recombinant DNA to pass through it. Similarly, the liposuction process mediated gene transfer are circular lipid molecules having aqueous interior that can carry nucleic acids. The nucleic acids are attached to the liposomes, and it is transferred. Direct transfer of DNA is transferred into by using microinjection, or we can say a gene gun, which leads to particular bombardment, and like a gun shoots, the desired DNA or recombinant DNA is transferred, entered to the cell. So when we talk about the application of recombinant DNA technology, the technology has a large applications like for diagnosis, molecular diagnosis, prenatal diagnosis. It is basically at the micro level. So as we talk about DNA, so it enables to detect a lot of things, like we can get the prenatal test before the birth of the child. We can know its genetic or genomic history. So, and diagnosis of highly sickle cell disease, genetic disease, familial hypercholesterolemia, cystic fibrosis, etc. Now, the modern gene therapy also involves the genetic alteration, that is recombinant DNA. We can alter the defective gene and remove it with a healthy gene. Beyond this, genetic modified organisms, genetic modified plants can be made, and this helps in our application in agriculture. So, friends, that was the basics and the high discussion when we talk about recombinant DNA technology and its application. Hope you like it. So be awaited for the next interesting video. Thanks.